This video is about PCR, or polymerase chain reaction, where the whole idea of basically this event is to double a strand of DNA. And what do I mean by that? I mean effectively you get a template strand of DNA, and through magic that we're going to talk about, you end up with two strands of it. So it is sort of DNA amplification, or it's fake DNA replication to be quite frank. And what you do is you put all this stuff here into a thermal cycler that I showed you before. So you put it into one of these little wells here, you can actually see them running, some here, and you let it do its magic. The process by which you do this is you firstly heat it up, and the reason that you heat it up is that you need to break the hydrogen bonds between the template strand that you have put in. So all those hydrogen bonds need to be broken because you need exposed bases. So in order to do that, in order to get those hydrogen bonds to break, you put it to 90 degrees centigrade. That process is known as denaturation, because similarly to proteins, you are breaking sort of internal intermolecular forces, in this case hydrogen bonds, between your sort of macroscopic molecules, or sorry, larger biomolecules, in this case the covalently bonded nucleotides. Then the next bit that people don't like, because this is not how you actually understand DNA replication to work, but you have to turn the heat down to allow for hydrogen bonds to reform, but this time not between the strands, but between a primer and the template strands. This phase is known as annealing, and annealing just means the sort of wide-ranging formation of hydrogen bonds. So you would say this primer is annealing to the DNA strands that you have. When I'm talking about primers, this is a thing that usually upsets hate because you've never seen it before. Basically, these are short little strands of DNA or RNA. You can actually use both now. And what they do is they allow for the DNA polymerase to sort of latch on to something. In terms of the DNA polymerase is very, very poor and actually can't grow from a single nucleotide. So the idea of not having a primer, the alternative would be to have a single nucleotide on its own binding to another single nucleotide on its own. It's really bad at doing that, and it can't do that. It has to start from a pre-made chain that you put in. So earlier on, when I showed you here, there were primers ready to go. In life, this is also true. So when I say in life, I mean sort of in semi-conservative replication, in the S phase of the cell cycle, you actually also need primers. You just weren't taught them before in lower sixth because they're not on the syllabus at that point. So this is true of everywhere. So you have to cool it down to allow these primers to anneal. And then you let the DNA polymerase do its thing, and you whack up the heat to 72 degrees for chain extension, or just extension. And this is where your DNA polymerase will whiz along and add in the new free nucleotides that were in there according to complementary base pairing. And if you notice here, the DNA polymerase very happily starts from where the primer was. So it's very happy doing it when it's got a primer in front of it. So this is where people start to think, what? Because that seems rather high for your DNA polymerase. And the answer is we use a very special type of DNA polymerase that comes from Thermophilus aquaticus, which is an archaea, I believe, that lives in hot springs. And it is very, very comfortable at these temperatures. It has a huge amount of disulfide bridges in its tertiary structure, and that allows it to maintain these sorts of temperatures, maintain itself in these sorts of temperatures, and allow the reaction to continue. And effectively, if you leave it at 72 degrees for a while, it will carry on, and you will end up with two chains. Perfect. You have got your perfect two chains that we originally started with. But this is a chain reaction where you continue to keep going on. So what you do is you get that mixture, you put enough in at the beginning that you can do a huge amount more cycles than this, and you heat it up again. So rather than having two this time, there'd be 4 strands of 2 DNA molecules. You denature them, you anneal them, and you chain extend them. So at that point you have 8, and then you do it again. So you've still got enough primers, Taq polymerase, and 3-deoxynucleotides to do it again. And this is where Taq polymerase comes in handy. So if this were done with a normal DNA polymerase, what you could do is add in DNA polymerase here and then have the chain extension step at, I don't know, like 37 degrees or something. That would work. But that's a real nightmare, because the next time you went to do the next round, so okay, you put it in, let it extend, you've got your 4 chains or your 2 chains, whatever you're talking about, heat it back up to 90 and you denature not only the DNA and break all the hydrogen bonds with it, you'll denature your enzyme and you'll have to put more in. If you use Taq polymerase, you don't have to do that. It's very comfortable at higher temperatures, so it stays in the solution. That's why Taq polymerase is so important. It doesn't denature at high temperatures, and it allows the chain reaction to go on without having to constantly add in DNA poles at this point. Problem is, that is not what PCR is only used for. So there's some clever uses of PCR outside of this. Well, not outside of it, but a little bit more subtle. So let's look at an example here where you've got someone who you want to know which allele they've got. So what you can do is you can put in a mixture with a primer that you've designed. So if you look at these 2 alleles, we don't know which one of them they've got, and we don't know which one of them the person's got. So what you can do is take a PCR sample. Let's say they're homozygous, just for the simplicity of this video. So whichever one they've got the same of it. Okay, and you say, right, I'm going to add in, I think they've got allele 1, so I'm going to add a primer that will only anneal at this point. So I'm going to design my own primer, and I'm going to say, right, it's going to be complementary to this. So if my primer is DNA, it'll be T, G, T, A, T, A, C, and I'm going to add that in. If I get amplification, I know they've got a copy of that, because otherwise, if it didn't amplify at that point, then you know that the primer is not sticking to it. The primer is not going to anneal, so you won't get any extension. So if I then go, right, okay, so I got some amplification, and I know they're homozygous, fabulous. Okay, I know they're homozygous for allele 1 because I got amplification. If you don't know that, and let's say this time they are heterozygous, what you can do is you can run it once with a primer that is complementary to this, and then run it again with a primer that is complementary to this. And if you get amplification both times, you know they're heterozygous. So it can be used to test for what genes, or alleles I should say, are present. Equally, it's actually what was used in a COVID test. So what you did was, effectively, COVID annoyingly uses RNA as its main nucleotide. So you have one extra step where you get the RNA and you turn it into DNA with an enzyme known as reverse transcriptase, which you will meet in genetic engineering later. So you make it into DNA, and then what you do, all the COVID test was, not your lateral flows, the ones you had to sort of send off for, which were a lot more accurate, is all you do is you put DNA in a PCR, that DNA you've made in a PCR machine with primers that are sort of for COVID genes, and if you get amplification, you know COVID was in the cells that you were swabbing, basically. And that's how the sort of original COVID test works. It was just a PCR test, effectively. And that's it. So the next video, or the next video I'd recommend watching, is, say we were to do a mixture where we put both of these primers in, so the primer to allele 1 and allele 2, and got a mixture of these 2 alleles. How could we separate them? And the answer is gel electrophoresis.